Okay, this is where we left off. So we got the food that you ingested into your stomach. We were talking about what happens in the stomach. And we were referring to the fact that gin means gives rise to, as well as reminding ourselves, I guess, that ASE is the what gives us the tip that something's an enzyme. And remember, an enzyme is something that acts on a product but doesn't get changed itself. And so what pepsinogen, it's converted to pepsin because of that low pH. So that's one of the reasons why hydrochloric acid is secreted into the gastric pits, the stomach, as well as the food that you ate could have an incredible amount of E. coli, other bacteria, salmonella, other things that that won't live in that acid, so the acid will kill it. So it's kind of a safety measure for you. And the other thing that happens is the acid acts on the bonds, actually tertiary bonds, turns proteins into long polypeptide chains. So the tertiary bonds. So remember, a protein starts out as a three-dimensional product that goes in, and that three-dimensional product then turns into a long, say, folded fan. That folded fan are what we consider polypeptides, and then they need to be separated one from the other because it's actually an amino acid that goes into the bloodstream. And that's all part of the process. Then digestion continues with this acid, this pepsin, this amylase, and this lipase. Then when after the food exits the stomach, goes into the small intestines, and generally, and remember, the first part of the small intestines is the duodenum, and particularly the duodenal ampulla is that first expanse of the duodenum, and it's chyme at that time. And what happens is that acidic fluid flows into the small intestines. Well, we're talking about seriously acidic fluid, so immediately it needs to be neutralized. So it's neutralized with mucus, yes, but with bicarbonate ions. So you have bicarbonate ions that are just there saying, I'm going to take every single one of those hydrogens that come through and lower that pH right away. The pH of the stomach is somewhere around 1.52. The pH that when it hits the duodenal ampulla needs to be more like, I would say more neutral, more like 7 or so. So that's how quick that happens. Liver makes bile, and this is bile. What it does is it actually surrounds big lipid macromolecules with what's known as bile salts. Those bile salts are a polar molecule. That polarity, remember, a polar molecule, you're thinking of something like water, where you have a big oxygen molecule and then two small hydrogens, where the electrons are not shared equally, so you actually have a negative portion of it and a positive portion. Well, these bile salts make a polar molecule that surrounds the fat. We call it a chylomicron, and that allows the fats to move into the lymphatic system because of that polarity that's attached to it. And then water is added and reabsorbed, so you have this, turns it in more and more emulsification. No, emulsification is not the right word. That's the word that we use with bile. More and more mixing with liquids. The digestion then, as I said, pH goes to closer to 7, and then what these enterocytes, these are endocrine type cells that are embedded in the entire GI tract system. We'll talk about it, but the absorption of building blocks through enterocytes to the liver. One of the reasons why that happens is things are absorbed into the liver, so then the liver can manufacture certain things. The liver manufactures your antibodies, for example, and the antibodies are protein based. So protein portions need to be absorbed into the liver. And then much digestion still needs to, well, actually, by the time everything gets done with the small intestines, it's going to move on to the large intestines. Remember, the large intestines is where we're getting ready to eliminate. So most of the digestion is done by the time it moves from the small intestines to the large intestines. And then in the large intestines, what happens is water is absorbed, so you have dehydration. Water is absorbed back into your system, compaction, and then elimination. And that's the story the second time through. So we'll talk about those processes even more. And again, hopefully you don't get bored, but you hear it enough that it stays in your head. Then the accessory glands, your salivary glands, they're going to release amylase. This is in the mouth. Start breaking down starch. Lipase to start breaking down lipids. The pancreas is considered an accessory organ where there's a whole menu of enzymes released. Bicarbonate ion is released, as well as the pancreas does release insulin and glucagon, and they are released in concert with digestion, because digestion ultimately creates an increase in blood glucose levels, but not to be confused. The pancreas is that organ that is an exocrine and an endocrine organ. Exocrine when it's working in the digestive system, endocrine when it's working, when it's releasing insulin and glucagon. So we'll talk about that later. I mean, particularly we'll talk about it a whole lot when we talk about the endocrine system, but please understand that insulin and glucagon are only listed here, but they aren't I guess directly part of digestion. They're part of what takes the glucose into the cell after it gets into the bloodstream. Liver makes the bile. Liver makes bile, stores much. Liver has, it depends. People can't agree on how many functions the liver has. Some people say 600, some people have 500. The liver has a lot of functions. It's a very hard-working organ you have in your body. Stores building blocks, amino acids, stores some of the building blocks with regard to blood proteins, and detoxifies drugs, so breaks drugs apart. Gallbladder stores and concentrates the bile from the liver. So it's just a sac that's actually going to store that bile, and in the storage basically creates a more concentrated form, and then contracts and releases it on cue. That's the story. So a little bit more anatomy. Most of the digestive organs are found in the peritoneal cavity. And you remember that from your first anatomy class where you learned the cavities of the body. The peritoneal cavity is in the abdominal cavity. It becomes the peritoneal cavity when these organs are housed in the serous membrane. Serous membrane, remember, is a two-layer membrane with fluid in between, no opening to the outside world. And these organs are housed in that membrane, kind of walled off from that which is on the outside, and actually the outside's walled off from them to a certain degree, as much as two layers of cells can do. But they do a pretty good job. And some of them most of them are housed in that serous membrane. Some are housed outside of the serous membrane, so we call those retroperitoneal. So posterior to the parietal peritoneum, parietal is the outer layer. Remember, the layer that's closest to the organs is called the visceral peritoneum. Parietal is the outer layer of this peritoneal sac. And inside, but they're actually embedded in connective tissue. And you have the pancreas, the duodenum. You can actually see it here where it's outside. The sac is actually the kind of the white bluish stuff. The other thing that you're seeing there in here is you're seeing the lesser omentum. The lesser omentum attaches the stomach to the liver. you're seeing a little bit of the greater omentum. The greater omentum hangs off the greater curvature of the stomach. You're seeing the mesentery. The mesentery is what they call the apron that actually the small intestines are suspended in the peritoneal cavity. So they're hanging there in a they're hanging there in what hopefully is a very orderly way, because the small intestines are constantly moving and we don't want them to move out of position. Here's another picture of the mesenteries. Mesenteries, think of the connective tissue that actually is loosely attaching all parts of the or most parts of the GI tract one to the other, and you do see the greater omentum there on the smaller curvature of the stomach, or the lesser omentum on the smaller curvature, the greater omentum on the larger curvature of the stomach, and then you see some of the mesocolon, the folds, and the mesentery proper with regard to the small intestines and large intestines. The part on the left is showing you the development of the GI tract, how it develops in the peritoneal cavity, and some of the things that you have are remnants of the development, maybe more important when it was developing. But it is very important that the contents of the GI tract stay walled off from other parts of the body and vice versa. Here you have a slide that's kind of scary, but sometimes it's good to have it written out. We're moving on to the next topic. We're going to do, we're going to go more microscopic with regard to the walls of the alimentary canal. And the walls of the alimentary canal, I might come back to this. I have a picture which is probably better. From deepest to most superficial, you have the mucosa, and by the way, that word mucosa should not be capitalized. We only capitalize anatomical terms when it's a proper name. So all these times when I'm capitalizing, if it's an abbreviation, it should be capitalized. So GI should be, but the other ones shouldn't be. They're just typos, and I need to take care of those sometime in the future. But deepest to superficial, you have the mucosa. It's the innermost layer. It's closest to the opening. It has a mucous membrane, so it's very, very lubricated. The lining, it's epithelial cells that can secrete mucus, and it contains glands that are self-regulated. Anytime you see this word intrinsic, it means that the regulation isn't systemic. It's regulation only in that particular area. And then you have this lamina propria, sorry. And what that is basically a barrier between layers. It tends to be charged proteins that repel things because of the charge. Loose connective tissue, and very rich in capillaries and lymphatic tissue, so lymphoid tissue. So you can see that this would be where things would be absorbed, where you have these rich capillary beds to absorb nutrients. We are in the innermost, deepest layer. Next layer is submucosa. You got to love that medical terminology. It helps you all the time. So that which is after the mucosa, I guess you might say. And that is connective tissue to help reshape the organs once the food passes. Major blood and lymphatic vessels, and some glands. Then you move on to the muscularis externa, 2 layers of circular and longitudinal muscle to contract, create peristalsis. The serosa, the outer layer, and that is actually one with the visceral peritoneum, that serous membrane sac, sometimes called the adventitia. And we need to stop there because I'm running out of time. We'll pick it up on the next one.